Neighbouring tissues must remain distinct and prevent their cells from mixing with each other. In some cases, adjacent tissues are separated by a physical gap, such as the one that forms between the ectoderm and mesoderm of early Xenopus embryos. This gap, known as Brachet's cleft, allows the mesoderm to move across but not into the overlying ectoderm. Rudolf Winklbauer and colleagues have previously demonstrated that Brachet's cleft is generated in part by F receptors and their ephrin ligands, which induce cell repulsion at the mesoderm-ectoderm boundary.
But the thing was that the receptors and ligands were not expressed complementarily. We found many receptors, many ligand isoforms expressed all over the place. So the question was why a cleft should form just at that position and the tissue not fall apart. All cells have receptors and ligands and repel each other.
This suggested that additional mechanisms must regulate the formation of Brachet's cleft. Winklbauer and colleagues, led by first author Olivia Liu, identified a key role for the mesodermal transcription factor Snail1.
Normally, even in the light microscope, at low magnification, you see a cleft between ectoderm and the mesoderm. And when we knock down Snail, then the cleft is gone in a certain region. And we could rescue it when we added Snail1 RNA to the knockdown morpholino, and from that we concluded it's involved in tissue separation at Brachet's cleft.
This phenotype could be recapitulated in vitro, as mesodermal cells lacking Snail integrated into ectoderm explants, whereas wild-type mesoderm remained separate. Moreover, with the help of Ashley Bruce's lab, Liu et al. Found that Snail was also required for the formation of Brachet's cleft in early zebrafish embryos. Through a series of knockdown and rescue experiments, the researchers found that Snail expression was induced by a non-canonical Wnt signaling pathway initiated by the Wnt receptor frizzled 7. But what does Snail do to promote tissue separation? Liu et al. Found that Snail enables the function of paraxial protocadherin, or PAPC, a mesodermal cell membrane protein previously shown to promote the formation of Brachet's cleft. Ectopically expressing Snail and PAPC in ectodermal cells was sufficient to induce their separation from wild-type ectoderm not expressing the two proteins.
When we injected Snail and PAPC on one side of the ectoderm and not the other, we got a cleft-like boundary now within the ectoderm. But what we found then is when we expressed Snail all over and PAPC only on one side, we still got the boundary. But when we expressed PAPC all over and Snail only on one half, the boundary was gone.
This suggested that tissue separation occurs at the boundary between PAPC expressing and non-expressing cells. Winklbauer and colleagues investigated whether they could see this effect at the cellular level, mixing ectodermal cells that either did or did not express a truncated version of PAPC, called MPAPC, that promotes tissue separation independently of Snail.
So like cells attach to each other and form lumps of cells. But the unlike cells, like MPAPC cells and non-injected cells, also attach to each other. They were still adhesive, but there was almost like a gap between these cells. And the other thing was that laterally the cells could glide past each other at these gaps.
Mesoderm cells, which naturally express PAPC, formed similar cleft-like contacts with PAPC-negative ectoderm cells in vitro. Knocking down PAPC abolished these clefts and caused the cells to adhere tightly to each other as if they were identical cell types. These cleft-like contacts might therefore reconstitute in vitro the structure and function of Brachet's cleft in vivo.
So we looked in the embryo, in the electron microscope, how does the cleft actually look like? And what we found is, surprisingly, that there were huge gaps between cells, but there were other regions where cells got in close contact, thinner than 30 nanometers. And the contact is so close that cadherins could reach across, but altogether less than, I think, 17% of the contact area was close contact, and the rest was different kinds of gaps. And then we did the same thing in cell pairs, and found that the same situation was observed here. We had close contacts, but mostly we had wide separation distance between the two cells.
These minimal contacts could allow mesoderm cells to remain separate from the ectoderm, but successfully attach and migrate across it. In the absence of PAPC, the number of close contacts was greatly increased. But how does PAPC promote cleft formation and tissue separation? One mechanism, Liu et al. Found, is that, in conjunction with Snail, PAPC promotes the removal of planar cell polarity proteins, such as disheveled 2 and prickle, from the ectoderm-mesoderm boundary.
Disheveled and prickle are in the cell membranes in the mesoderm, but we found that they were strongly reduced at the boundary to the ectoderm. And when we knocked down PAPC, the number of these puncti of disheveled or prickle increased strongly at the boundary. It became similar to the level within the tissue.
Knocking down the planar cell polarity proteins decreased cell adhesion and rescued tissue separation in embryos lacking Snail. Planar cell polarity proteins may therefore enhance cell adhesion and counteract ephrin and F receptor-mediated cell repulsion within the mesoderm. But the proteins are downregulated at the ectoderm-mesoderm boundary by PAPC and Snail, weakening cell adhesion to permit repulsion and tissue separation. However, experiments suggest that PAPC plays an additional role in cleft formation besides the downregulation of planar cell polarity proteins. Winklbauer and colleagues now want to determine what this other mechanism is, and to investigate how PAPC-expressing mesoderm cells distinguish each other from PAPC-negative ectoderm cells. In the meantime, however, you can learn more about how PAPC mediates self-non-self distinction during Snail-1-dependent tissue separation in the paper by Liu et al., published in the March 16, 2015 issue of the Journal of Cell Biology.
You